We're still in chapter 24, and we are going to talk about the oxidation of glucose. So glucose is catabolized via the following reaction. And I'm trying to get my cursor. There we go. It is glucose, or C6H12O6, plus oxygen, O2, is going to give us water, H2O, carbon dioxide, CO2, ATP, and heat. So normally when we look at a reaction, which is what this is called, this is a reaction, we have the reactants on this side of the arrow and the products on this side of the arrow. This means that glucose and oxygen are going to give us water, carbon dioxide, ATP, and heat. Now you may notice also that there's some numbers up here, 6, 6, and then another 6. And the reason for these is the fact that this equation has to be balanced, which means that we have to have the same number of carbon on this side as we have on this side. Same with hydrogen and oxygen. So complete glucose catabolism requires 3 pathways: glycolysis, Krebs cycle, which we're also going to call citric acid cycle, and electron transport chain and oxidative phosphorylation. So this diagram is a little scary, but I'm going to do a little overview. Okay, sorry if that little arrow was just circling around, I didn't even see it. Okay, we have glycolysis occurring out here in the cytosol. Okay, and with glycolysis, and we're going to talk about all this in a lot greater detail in this particular video, glycolysis is going to be changed to 2 pyruvic acid molecules. So one glucose is going to get split and converted, and a bunch of stuff's going to happen, and we're not going to get into all the details, but it's going to start off with glucose, and we're going to end with 2 pyruvic acid molecules. And then pyruvic acid is going to enter the mitochondria, where it's going to go into the citric acid cycle, also known as the Krebs cycle. During the citric acid cycle and in glycolysis both, we're going to produce ATP via substrate level phosphorylation. And we talked about that in the very last video. And very few ATP are made this way. Now, in addition, chemical energy is going to go from glycolysis and get sent with via high energy electrons to the electron transport chain and oxidative phosphorylation from both glycolysis and the citric acid cycle. And then during this process, we are going to produce a bunch of ATP via oxidative phosphorylation. So glycolysis is also called the glycolytic pathway. It involves a 10-step pathway. It's anaerobic, which means that it occurs whether or not whether or not oxygen is present or not. Glycolysis occurs in the cytosol, and again, glucose is going to be broken down into 2 pyruvic acid molecules. And there are 3 major phases of glycolysis, but that's not important for us. What I want you to see is we have glucose starting up here, 6 carbons. We have some ATP that's being used. We have some ATP that's being made. And in the end, we have 2 pyruvic acid molecules. So although 4 ATP are made, I showed you those 2 in the beginning that are needed to prime the system. So they need to be subtracted. The final products of glycolysis are 2 pyruvic acids, 2 reduced NADH+, and a net gain of 2 ATP. For glycolysis to continue, more NAD+ has to be present to accept more hydrogen atoms. And the supply of NAD+ is limited. So once the NAD+ becomes NADH, that NADH has to go and donate its accepted hydrogen atoms to become NAD+ again, so it's free to pick up more hydrogen atoms so glycolysis can continue. If oxygen is present, NADH will transfer its hydrogen to proteins in the electron transport chain, and that occurs in the mitochondria. If no oxygen is present, NADH will give its hydrogen atoms back to pyruvic acid, reducing it to lactic acid. And you may have actually experienced this if you've ever been working out, and let's just say you decided that you want to work out to exhaustion. Okay. At some point, you're going to start to feel the burn, and the burn is lactic acid. So the fate of lactic acid, some may leave the cell, some may be picked up by the liver, where it can be converted back to glucose 6-phosphate, and then some may be oxidized back to pyruvic acid when oxygen becomes available. At that point, it can enter aerobic pathways. Now, prolonged anaerobic metabolism can lead to acid-base problems, which makes sense because you're making a bunch of lactic acid. Now, glycolysis results in faster ATP production than aerobic respiration, but it yields far less. So I just showed you that the net gain from glycolysis is 2 ATP, and we're going to find out later that oxidative phosphorylation is actually going to yield 28 ATP, about. This is the citric acid cycle, is also called the Krebs cycle. It occurs in the mitochondrial matrix, and it's fueled by the pyruvic acid from the glucose breakdown, and also, which we'll talk about a little bit later, fatty acids from fat breakdown. Pyruvic acid must be actively transported into the mitochondria because it's a charged molecule, and once inside the mitochondria, the pyruvic acid is going to enter the transitional phase. The transitional phase is where we're going to convert pyruvic acid to acetyl-CoA, or acetyl coenzyme A, and this is going to occur in 3 steps. The first step, 1 carbon from the 3-carbon pyruvic acid is going to be removed, and it is going to produce carbon dioxide, which is going to diffuse into the blood and then be expelled by the lungs. The next step is oxidation, the remaining 2 carbon fragments. So remember, pyruvic acid's 3 carbons, we take out 1, so now we have 2. It's going to be oxidized to acetic acid by the removal of hydrogen atoms, which are going to be picked up by NAD+. And then finally, we have the formation of acetyl-CoA. Acetic acid then combines with CoA to form acetyl-CoA. And what we're going to focus on is this part up here. So we have the pyruvic acid from glycolysis is coming in. We're going to kick off a carbon. We're going to make some CO2. NAD plus is going to come in here, pick up hydrogen, and that is going to convert it to acetic acid. We're going to join it with CoA, and we're going to form acetyl-CoA. Coenzyme A then shuttles its acetic acid to an enzyme in the citric acid cycle that joins it to the 4-carbon oxaloacetic acid, and this is going to produce a 6-carbon citric acid, and this is why this that Krebs cycle is also called the citric acid cycle. So during the citric acid cycle, there are going to be 8 steps. The original acetic acid is going to be decarboxylated and then oxidized to keto acid intermediates. You do not have to know what those keto acid intermediates are. And this is going to result in NADH plus hydrogen and FADH2 and CO2. In the final step, oxaloacetic acid is regenerated. In the citric acid cycle, for each turn, we're going to get 2 CO2 from decarboxylation, 4 molecules of reduced enzymes, 3 NADH, and 1 FADH2. I'm just going to abbreviate that NADH, so we're just going to call it NADH instead of NADH and hydrogen, and 1 ATP. Now, it's important to remember that we're going to start with 2 pyruvic acid molecules, because if you remember from glycolysis, glucose is at 6 carbon, it splits into 2 pyruvic acid molecules. So for each glucose, we're doing citric acid cycle twice, 2 turns. So the final amount, the amount of final products from one glucose is going to be doubled. So it's going to give you 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATP. Once we add the products of the transitional phase, the final products for the breakdown of one glucose are 6 CO2, which, if you remember, that's part of your equation, 8 NADH, 2 FADH2, and 2 ATP that are made via substrate level phosphorylation. So let's look at this. We have acetyl-CoA coming in, joining with this 4-carbon molecule, making citric acid, and then we're going to different intermediates, and as we go through these intermediates, we have things happening. We're kicking off the CO2 here. We have NAD plus coming in here, getting reduced to NADH. We have more of that occurring here. We're making the ATP via substrate level phosphorylation here. Another CO2 is kicked off. Here's where the FAD comes in and is reduced to FADH2, and then another NAD. And then we just start back at the beginning. I lost my cursor. That is not what I was trying to do. There we go. And then we start right back where we begin. I'm sorry, we end. Yes. Anyway, we come back. We go full circle. So NAD must be oxidized to ensure that NADH must be oxidized. It's very important. NADH must be oxidized to ensure that there is plenty of NAD so glycolysis, transitional phase, and citric acid cycle can continue. And that is going to occur in the electron transport chain. The breakdown products of fats and proteins can also enter the citric acid cycle at certain points to be further broken down. And also, cycle intermediates can be used as building materials for anabolic reactions rather than just catabolic reactions. And then we're going to talk about electron transport chain and oxidative phosphorylation. So oxidative phosphorylation consists of 2 phases. We have phase 1, which is electron transport chain, and phase 2, which is chemiosmosis. In the electron transport chain, we are going to create a proton gradient across the mitochondrial membrane using high energy electrons that are removed from hydrogen from food fuels. Chemiosmosis is going to use the energy of the proton gradient to synthesize ATP. So I just want you to know kind of a general, let me blow this up some. Okay, that's better. So we have NADH plus this hydrogen here, this hydrogen ion, and FADH2 coming in, and they're going to drop off their electrons. And as they drop off their electrons, I lost the cursor again. There we go. As they drop off their electrons, protons are going to be pumped across these membrane spaces. And these electrons are going to kind of go from one of these complexes to the next complex to the next complex. And again, as it's moving along, protons are going to be pumped through the membrane. This is going to set up a proton gradient. And then the chemiosmosis, which is phase 2, is going to use this proton gradient to synthesize ATP. And we're going to talk about specifically how this works in another slide. So electron transport chain is the only pathway that directly uses oxygen. And it uses oxygen as a final electron acceptor. The citric acid cycle will not run if oxygen is not present, but it does not directly use oxygen. And then as an overview, NADH and FADH2 from glycolysis and Krebs deliver hydrogen atoms taken from glucose to electron transport proteins. The electron transport, electron transport chain proteins. The electron transport chain will use the energy harnessed in electrons of hydrogen to pump protons across the membrane to create a proton gradient. Some of the hydrogen ions will combine with oxygen to produce water. And I should also note, if I haven't before, hydrogen ion and proton is the same thing. It just, sometimes we call them hydrogen ions and sometimes we call them protons. It just depends on the particular subject that we're discussing. And we've already seen this diagram. The one thing I didn't point out, and that I would like to, is this right here. This is where the oxygen, we have 2 hydrogen ions combining with half an oxygen molecule to form water. All right, phase 2, chemiosmosis. And so these are the steps of ATP synthase, production of ATP. So the first thing is the protons, or the hydrogen ions, are going to flow down the gradients through the stationary stator of ATP synthase. As they pass the rotor mechanism, they bind to one subunit, causing it to change shape, and it's going to spin. The spinning rotor is then going to turn a connecting rod, and as that rod spins, it's going to activate catalytic sites on the knob that's going to join a high energy phosphate with an ADP, forming ATP. I did not ask questions about specifics for chemiosmosis on the exam. But we have, I'm going to go over this again. We have protons are going to flow down their electrochemical gradient through the stationary stator. Okay. Through the stationary stator. Then each proton is going to bind to a subunit of the rotor, which is going to cause it to change shape, which is going to cause the rotor to spin. That spinning rotor is going to turn the connecting rod, and then as the rod spins, it activates the catalytic sites in the knob that's going to join that high energy phosphate to ADP to make ATP. And these are some energy converting ATP synthase rotor rings. ATP synthase works like an ion pump in reverse. The proton gradient also drives the energy needed for ADP, pyruvic acid, inorganic phosphate, and calcium to cross into intermitochondrial membrane. And then a summary of ATP production. We have the flow of energy. I don't know that I like necessarily this flow of energy. Don't get too hung up on this. Glucose to NADH plus a hydrogen is then to electron transport chain, proton gradient energy, and then to ATP. Because it's also FADH2 is in there. And anyway, don't get too hung up on that. Net energy gain from the complete oxidation of one glucose molecule. So we're going to make 4 ATP from substrate level phosphorylation. 2 are coming from glycolysis, 2 are coming from the citric acid cycle. And then we're going to make 28 ATP via oxidative phosphorylation. So for each NADH that's brought in, the proton gradient is going to generate 2.5 ATPs. And so when we multiply the 10 NADH by the 2.5, we get 25 ATPs. For FADH2 brought in, only 1.5 ATPs are created. 2 FADH2 are made, so 3 ATP are created. Totals between substrate level phosphorylation and oxidative phosphorylation are going to give us 32 ATP, but there is energy required to move NADH generated in glycolysis into the mitochondria, which is going to use about 2 ATP. So the final total is 38 ATPs produced. But I'm going to tell you that the Final total is nobody really knows the answer because I learned 38 and then they said 36. And then when I started teaching, it was 34. And at some point it went down to 30. So it really depends on the class, the textbook. It depends on a lot. Ooh, complicated diagram, but it's really not that bad. So let's look at it in little chunks. We have glycolysis, and in glycolysis, we're taking glucose and we're going to convert it to pyruvic acid. We are going to make 2 ATP via substrate level phosphorylation, and we are also going to send 2 NADH to the electron transport chain and oxidative phosphorylation. This pyruvic acid is going to enter the mitochondria and go into the transitional phase, which is not really marked too clearly here, but that's what this is trying to show us. And 2 NADH are going to come off of this and then once again go to the electron transport chain and oxidative phosphorylation. From the transitional phase, we make 2 acetyl-CoA. The 2 acetyl-CoA are going to go into the citric acid cycle. And in the citric acid cycle, we're going to make 2 ATP via substrate level phosphorylation. And we're going to send 6 NADH to FADH2 to electron transport chain and oxidative phosphorylation. And then we have our totals here from all of the NADH and the FADH2. And this is going to give us 28 ATP by or via oxidative phosphorylation. When we add the 2 and 2 and 28, we get 32, but we have to subtract the shuttle cost of 2 ATP to give us about 30 ATP from a glucose molecule. Here are your questions. That is all I have for you for this video.